During mitosis, the cell nucleus divides into two daughter nuclei that are morphologically and genetically equal to each other. Each new nucleus has the same set of chromosomes as the parent cell. The duration of the mitotic cycle averages from 10 to 50 hours. Then, the cytoplasmic part of the cell divides, forming two daughter cells, each receiving one daughter nucleus. The new cells are similar in structure and function, both to the parent cell and to each other. The two daughter cells, each approximately half the size of the original mother cell, begin to grow and perform their functions again. In multicellular organisms, mitosis determines the growth of the plant body, which is why it is often called somatic division. Somatic cells have a double set of chromosomes. Reproductive cells have a single set of chromosomes. Mitotic nuclear division occurs similarly in the vast majority of organisms. The main events of the mitotic cycle occur during the reduplication of chromosomes and the completion of DNA synthesis, during the doubling of mother cells' hereditary material, and then its even distribution between the daughter cells. The biological significance of mitosis is that it ensures the formation of genetically equivalent nuclei in each new cell. Mitosis proceeds through several stages. The majority of cell cycle falls on interphase, and a smaller part falls on the processes of mitosis itself and cytokinesis. At this stage, the cell prepares for division. It grows, the genetic material DNA duplicates, many necessary substances are synthesized, structures required for mitotic division are formed. The doubling of the hereditary material DNA occurs as follows. The DNA double helixes separate into two strands, and next to each strand, a corresponding strand is formed based on its composition. During DNA reduplication, the biological information encoded in the genes is copied, and the organism's traits are passed on to the next generation. Reduplication is the basis of heredity. If you look under a microscope at a cell in the interphase stage, in its nucleus surrounded by the nuclear envelope, you will see the chromatin substance and one or more nucleoli. At the beginning of prophase, the chromosomes begin to condense and become visible as thin threads inside the nucleus. Each chromosome already consists of two sister chromatids connected by a centromere. By the end of prophase, the nucleoli disappear and the nuclear envelope breaks down into fragments. In the cytoplasm, the formation of the division spindle begins, composed of microtubules. At this stage of cell division, the chromosomes are finally separated. Chromatids begin to separate from each other, remaining connected only in the centromere region. Microtubules consist of specific proteins, forming a number of threads located between the poles of the nucleus like a spindle. Chromosomes attach by their centromeres to the spindle fibers and move along them to its equator, where they assemble in one plane, the equatorial plate, at the site of the former nucleus. Each centromere splits in half, and each chromosome divides into two independent chromatids, which become daughter chromosomes with their own centromeres. With the help of spindle fibers, the daughter chromosomes gradually shorten and move toward its poles. By the beginning of telophase, the daughter chromosomes reach the poles of the nucleus, the spindle disappears, the chromosomes swell, elongate, and gradually become indistinguishable again, taking the form of chromatin threads. Two new nuclei form. Simultaneously, nucleoli and nuclear envelopes appear around the two new nuclei. The chromosomes become invisible and each nucleus enters interphase. The second half of the chromosomes is completed in the nucleus during the interphase stage. After mitosis, the division of the cell itself, or cytokinesis, occurs. During cytokinesis, a middle lamella consisting of pectin substances forms between the two daughter cells. Initially, the middle lamella is disc-shaped, growing towards the walls of the mother cell by completing the cellulosic wall of the new cell. A construction forms in the cell's cytoplasm, and the organelles are evenly distributed into the forming halves. As a result, two new daughter cells are formed, each approximately half the size of the original mother cell. The daughter cells begin to grow and reach the size of a mature cell. Each cell has a nucleus with a diploid set of chromosomes. The cells can then divide again, undergoing a series of cell cycles, or begin to specialize in performing a specific function within a tissue. Gradually, they lose the ability to reproduce.